maybe we should make a start. I'd like to introduce James, which is a particular pleasure. It's not a pleasure that he's not here in person. We can
next time
sit and chat, but hopefully we should repeat this and make it more of an event. So James is a Londoner by birth and early childhood. He got hooked on computers really early on in his life, with one of those really early home computers that many of us played around with when we were young. And that at the same time, also as a child, I think he started to become interested in biology. So he then went to Oxford to study biology at a very particular time when developmental biology actually was really buzzing at the time. I was there then, and I can't remember quite whether James was attended our Tuesday club, which we used to have in your building. We called it the Tuesday Club, which is we met on Tuesday lunchtime for talks of developmental biology by junior people without slides, and it was really great fun. And that ran through from 1985 to 1993 or so. So anyway, so that was in the zoology department where you were. And so then after that, he went to do his PhD at Mill Hill in IMR with Rob Krumlov, where he worked on Hox gene regulation. And then shot off to Chile to work with Roberto on Xenopus BMP signaling and neural crest induction for a relatively short time. And then he returned to the UK to Edinburgh this time, first as a postdoc in the MRC. It was still called MRC Human Genetics Unit in Edinburgh, and then became a group leader in the same place. And during that time that he was in Edinburgh, he started to work on limb development and the like. And among the things he is very famous for is inventing a machine called the OPT scanner, optical projection tomography. We actually have one in our lab. And it's a brilliant machine. You can take an embryo, like a big embryo like this, and you can stick it in the machine. It'll rotate it, and you get 3D images and from it by using a tomography kind of algorithm. So this is his early computational skills really came to fruition this way too. I think actually there are two in the department.
Wow.
And so he must have got a little bit of money from us. So anyway, so he continued to work on limb development and the like, and the Spanish language and culture and the like, I suppose Catalonian as well, attracted him over to cross the channel down to Barcelona way back in 2006 or so, where he became a group leader there for a while in the big round building. You might see a picture of it later, perhaps in your slides. And 5 years ago was appointed as founding head of the new unit of EMBL, an outpost of EMBL in the same building, which focuses on tissue biology and disease modeling, which is quite a broad remit. But it's so James is now recruiting people with quite interesting, diverse skills to explore new avenues in that direction. Again, bringing together the biology and the sort of computational modeling, systems biology and the like. I've always been a fan of James because we share an interest in understanding regulatory logic of biological problems, which I think is kind of the same thing as, again, how the computer in biology, how does that work? Rather than one gene, one function, how does it all fit together, and where is the program that drives biology, not just in development, but beyond? And I think his talk today couldn't be more appropriate for the department because it addresses a question that was first articulated by none other than Lewis Wolpert, as you can see his title over there, way back in 1968, which was called the French flag problem originally. So, James, over to you.
Thanks very much. Thank you. So you can hear me okay, can you? And just to check, can you see my mouse moving around? Just because that's helpful if so I can point to things. Well, thanks very much, Claudio, for that introduction. In fact, no, I didn't attend your Tuesday clubs, was it? I mean, I was an undergrad and really just learning about development, but indeed it was an amazing time there, because I mean, the people who were teaching me, I mean, there was Fillingham, and there was Julian Lewis actually teaching, and Peter Holland, and you know, it was obviously the thing to do. So that's how I got into it back in those days, indeed. So the title is The Wolpertian French Flag, What's the Problem? Now, I'm only going to show one slide about our actual research. And so this is a lot of stuff kind of summarized extremely briefly, but we work on limb development. And in particular, we have been trying for ages to do many different things to try to sort of recreate the dynamical processes or the logic processes, as Claudio was saying, that govern this into a computer model, like a mathematical model or computer model or simulation. So that involves a lot of data collection, a lot of quantification, morphometrics, and ultimately, if this is gonna, just trying to rerun that movie. Sorry, until I haven't been giving that many presentations recently. A simulation. So this is actually a little snapshot from the kind of models that we're working on at the moment, which is a kind of agent-based model that could almost represent every single cell in the limb bud. In the mouse limb bud, in the time periods we're talking about, there are quite a lot of cells, like maybe 200,000 or 300,000 cells. And we are building in, you know, the little gene circuits that we believe are responsible, and putting signaling centers and the whole thing. And then the gradients of molecules such as morphogens can then orient cellular behaviors locally, like integration or migration and things like that, and try to put the whole thing together and see if it actually works, which is a bit like, what is our knowledge worth? We have a lot of papers collectively, you know, describing how the limb works, but if we put all those ideas together, does it actually make a limb bud? And I guess all I can say is that we're still working on that, and it's taking a very long time. But that is sort of in a nutshell, what we really work on. Now, I actually want to talk about just one specific thing that is not really what we work on, but I think it's kind of interesting. And in a way, you know, I was sort of reminded about thinking and talking about this because of course, the death of Lewis Wolpert earlier this year. And Claudio mentions, like, unfortunately, I'm not there physically in person. In principle, in a year, there will be this meeting, a 2-day symposium to celebrate the life and work of Lewis Wolpert. And in principle, I'll be able to come and speak at that meeting. But I thought anyway, today, it would be nice just to focus on one particular thing that I find fascinating, but it's almost a sociological thing about science, and what he actually said and what he meant and how people have interpreted it subsequently. So the first time I really started putting this together was, you know, a couple of years ago, which you will remember was touted as and was the 50th anniversary of positional information, because of his paper from 1969. And this has always been very much connected to this other phrase, the French flag something. But what exactly is the connection? And I was pointing out back then, in the celebrations of the 50th anniversary of positional information, that in fact, for the French flag, it was the 51st anniversary, because that phrase was published 1 year earlier in 1968 in this, which was in fact a chapter in a book, and is not as accessible, and it's quite understandable that not many people have seen the original one. Nevertheless, this was the original one. This was when the French flag problem phrase was first published by Lewis Wolpert. It was mentioned subsequently in this more famous paper a year later many times. But let's go in a little bit deeper about what this all means. Now, in a way, what I want to do is surprise you, maybe. about what you think you know about what he wrote and what he meant. And so let's start with the phrase positional information and the kind of concepts that are popularly associated with positional information. I would say that we often think of gradients, and in particular of morphogen gradients. We think of the idea of a spatial axis and then an axis of something else like a morphogen. And we think a lot of thresholds, threshold responses. There's one threshold at one concentration, there's another threshold response. And that can lead to cells making different choices if they have respond differently to different thresholds. So this is another important concept. We also associate this often with a phrase that we might call the French flag model. In fact, it's not just association. There are many papers with this written. And as a concept, of course, positional information, it's a lot to do with, let's say, what we might call a spatial ruler, the idea that there's sort of some monotonically changing thing across space, and cells make these decisions based on some idea of where they are. And we may contrast this with another idea, an idea that we've worked a lot on in the lab, on Turing patterning. And I'm going to sort of illustrate this now, but it doesn't really form much of this talk. I'm just doing this for completeness sake, which is to say, Turing systems, if you have the right molecules interacting in the right ways, activating and inhibiting each other, and then diffusing in the right way, then what you can get is something like this, where if you have lots of cells where this reaction exists and the diffusion is set right, spontaneously you can get molecular states that are non-uniform, breaking of the symmetry, and a kind of a pattern. And the important distinction here, which is useful in defining maybe what positional information means, is that here we've got some cells that switched on a gene, and here they didn't, and here they did, and here they didn't. But it's not because of, in some sense, the cells knowing where they are. They don't know where they are. Whereas here decisions are made, and in some sense, the sense of positional information, it's because the cells have information to let them know where they are in the grand scheme of things. So these are the ideas that we associate with positional information. It's also slightly the opposite of this kind of scenario. So there are also sort of criticisms that have been made about positional information, or caricatures in a way. I've put a few here. So this is just one quote, and there are many papers saying things like this. He formulated a simple toy model, the French flag, to demonstrate why spatial aspects need to be taken into account. That's correct. In this model, a substance is synthesized and released at one end of the tissue, the source, and diffuses to the other end of the tissue, where it becomes degraded, the sink. This results in a linear gradient of morphogen concentration across the tissue. Now, this is written in this paper, which I'm going to go into more detail. But in fact, this idea here was not called the French flag model, as you'll see. Another quote here is that a second problem, this is in relation to other problems, that the French flag model has, is that it's entirely static and feed forward. It does not consider dynamic aspects of pattern formation at all. Now, in fact, it's funny that this kind of criticism has been raised, because my interpretation really of what he was discussing here was neutral on the issue of dynamics. And in fact, in the paper, it does actually say, in the second paper, he does actually refer to a dynamical equilibrium, which can only mean that you're taking into account the idea that a system is dynamic and it will not immediately just be at the correct state. It takes some time and it goes through some transient dynamics. So this is interesting. All kinds of sort of criticisms have been thrown at the idea. And I suppose, you know, just to give you a little bit of my own personal background on this, because I've spent so many years working on Turing patterns and trying to reveal as much evidence as possible for the idea that fingers were patterned by a self-organizing Turing-like system rather than positional information. A lot of people have assumed that I was rather negative about positional information and gradient models. And I actually want to point out that that is not the case at all. And in fact, many of the criticisms that have been written about positional information and gradient systems, I want to now, defend his papers against those criticisms, because I think they were unfair criticisms. Another thing that's been written, and you may have read or you may have thought, is this: a third problem concerns size regulation. It only works for linear gradients, but fails for more realistic exponential gradient shapes. I'm probably not going to go into a lot of detail on this point, but this is not true depending on how you mean it. And it's also not true that Lewis Wolpert only talked about linear gradients in his papers. Yes, that he didn't only talk about linear gradients. He talked about exponential gradients as well and how they would work or would not work. So this is to a large extent a defense of Lewis Wolpert's work and pointing out how subtle and interesting it really was. So this is the second paper again, the more famous one. And he refers to the French flag problem, not the French flag model. And he says pattern regulation, which is the ability of the system to form the pattern even when parts are removed or added, and to show size invariance, as in the French flag problem, is largely dependent on the abilities of cells to change their positional information and interpret this change. So this is when he brought in really positional information as a concept, but linking it back to a problem in the previous paper. So let's go back to the previous paper, this chapter. Here, for purely sort of historical reasons, is the first real mention of the French flag in the literature of developmental biology, which is in this paper. I mean, it's in the title, and this is really where it is first mentioned in the actual paper. Now, because it's quite a long time ago, they printed everything in black and white, and it was all very simple. So here you just have B, W, and R representing blue, white, and red, the organization of the French flag. Now, I think, and this is kind of at the heart of what I'm trying to say, and I'm hoping not to sort of waste too much of your time on just this one point, because I'm laboring it a lot, and I'll try to get through it in a reasonable time. But I do think it's quite kind of fascinating, because a flag is a genius metaphor. And why is it a genius metaphor? It's not just a metaphor. It's a genius metaphor because of what I'm trying to illustrate here. I mean, this is a French flag, and this is a French flag, and this is a French flag. Hopefully you can see that they are dramatically different sizes. The whole point about a flag, the French flag, or any other flag for that matter, is that it is scale invariant. By definition, the concept of it, it is a pattern. It's like the most pure example that you can think of what developmental biologists would worry about with regard to pattern proportionality rather than size. It doesn't matter what size it is, it's still a French flag. If it's blue, white, and red, it's a French flag. Also, it doesn't matter how you make it. So neither the size of it nor how it is created is relevant for it being a flag. In this case, the perception of a French flag is because of different colored lights on the side of a building. And here, this is created whichever way, painting some fabric, and this might be coming from a printer. Neither the size nor how you make it is relevant for a flag. This was the key thing. And this is, getting to the point that I, found frustrating when I discovered this, that the phrase has become associated with a mechanism, with how you make something. But a flag is not. A flag is a set of colors in a certain proportions. So this is again part of the paper where he's trying to define the problem, that you have a number of units, which we can consider to be cells, but he was being quite abstract, a number of units, and they can do certain things, they can communicate in certain ways, and they've got to create a spatial arrangement flag. And in fact, it's also worth pointing out that even in this very first mentioning of something that would then continue to be discussed for another 5 decades, he mentions here, for a more difficult problem, consider the 2-dimensional case of the Union Jack. That would indeed be pretty complicated. Luckily, he sticks to the French flag, and I think fairly obviously this is because it's got only a certain degree of complexity, and it's essentially a 1-dimensional pattern, not a 2-dimensional pattern. Now, let's see what he says about solving the French flag problem back in 1968. From our attempts at the French flag problem, there seems now to be 2 main ways for solving it. 2 main ways. What are these 2 main ways? Well, there's a method that will be called the fixed gradient type. And there's another method, which we'll come to in a moment. The second type of model, the unit, meaning the cell, cannot compute where it is in the system. This is literally the definition of the opposite of positional information, which is what the French flag model is associated with. So model 1, fixed gradient method. And here we have this maybe first drawing of this idea that is replicated in papers and textbooks over and over again since then. And it is very well known to all of you, so I won't go over that. He never wrote anywhere French flag model in association with this idea. It's just simply not written. Then there was a second model in that very first paper called a balancing model. Now, the balancing model is very, very different. It does have a kind of a boundary condition, which is already non-symmetric. So there is something different about one end of the field than the other end of the field. And it's important to acknowledge that. But nevertheless, how does this method work? The idea is that the blue cells secrete a certain substance and white cells secrete a certain substance. And a cell that is on the boundary, like a cell that can detect both a blue cell on one side and a white cell on the other side, can switch its state. And it will switch its state based on the local amounts that it senses of the substance from the blue cells versus the substance for the white cells, which incidentally can diffuse very fast or fast compared to the rate of the system as a whole. So there's a production of this blue substance at a certain rate, and that promotes the switch from blue to white. So that cell becomes white. And that will continue and continue until there's enough substance from the white cells to balance the substance from the blue cells. This is why I called it a balancing model. Now, this can make a pattern like that. This is a simple way to illustrate it with just two colors. You can control where this boundary is. Like if the 2 production rates are equal, it'll be equal. But if you change the production rates, you can change the proportions so that the blue region will always be half the size of the white region. And you can introduce actually a third state and a third chemical to make the French flag. Now, the thing that I think is surprising, and I expect and hope that this is surprising to most people, is that in, again, this very first ever paper about the French flag problem, he already wrote, this type of model may be contrasted with the previous one, this solution to the French flag problem. Not only can a cell never compute where it is in the system, so there's no position information, but the system has no gradient. The latter point is of great importance, as it shows that gradients are not required for pattern regulation. Now, he's not saying that real systems will not use gradients. This is a very theoretical paper, but I think it's been completely lost how maybe sort of insightful and broad and theoretical it was, because in this very first paper, he promotes, two different ideas and says that, they would both be capable of solving the French flag problem: a gradient model and a balancing model. So, what's happened since then? I mean, what's been going on? So let's summarize the situation so far. In terms of models, he proposed two models: a morphogen gradient model and a balancing model. In terms of problems, he discussed one problem: the French flag problem. The problem, the French flag, was not a model; it was a problem, and we have to distinguish between problems to be solved and different hypotheses of how you solve them. The French flag problem was essentially a great metaphor for regionalization and scale invariant regulation. So that's what I'm trying to emphasize here. I think what his paper did, and I'll mention it a few more times, is provide a great metaphor for distinguishing between models and problems. Historically, afterwards, it seems that that's exactly not what happened in the field subsequently. It's also worth pointing out again that these two different models are sort of fundamentally different. They're not just slight variations. This is a positional information model, and this is a non-positional information model. So I also want to just focus a little bit on the second paper, which isn't in a way the main focus of my point, but I think it's a fascinating paper, which also is maybe not fully understood what was written in it. Because there have been many questions about positional information over the years. One is it simply a tautology? Like, is it really meaningful? Does it really help, or is it just sort of almost like a statement of the description? If you see that cells in different places can do different things, then that means almost by definition that cells in different places know that they're different. So what's really the value of this? Or is it really useful testable model? Is it a useful distinct category of mechanism? Hopefully I've already shown that I think it's a distinct category from more self-organizing. Or is it a call for more clear thinking in development? I'm going to skip over that statement, but I'm going to read out this one from his paper, because I want to also highlight in a way the modesty in his work, given that in a way he's known as like one of the dominating kind of thoughts in the field. He wrote in this paper, while the idea of position determining cell behavior is well known, the implications of this and the possibility of effectively establishing a coordinate system have not previously been explored. In a sense, the ideas put forward are relatively simple and do little more than redescribe known phenomena in new terms. I think it's kind of important to remember that this is how he described his own work there. Now, another point about this paper, going back to a criticism that I mentioned earlier, this is a figure from the paper. He was not thinking primarily of just linear gradients or gradients in one particular direction. He discussed all kinds of things in much more detail, non-linear gradients. And he frequently refers in this paper to an idea by Goodwin and Cohen on a phase gradient of two out of phase oscillators and traveling waves. This is an idea that was indeed published in the same year, and from what I understand, I think in the Lewis Wolpert chatted to Brian Goodwin in the taxi going to the conference in 1969 about this, and he was very keen on this idea. He mentions it multiple times during this paper. So the idea that he was primarily thinking of concentration gradients and thresholds to detect concentrations, it may have happened, it may have become true as the decades went by, but in the original idea, this was absolutely not the primary idea. He said it could represent, for example, the concentration of a substance, the phase difference between two periodic events, a particular state of the membrane, or a particular combination of molecules. He was very open about what this positioning information could be. It was not just morphogen concentration gradients. Neither was it only just one-dimensional. He does discuss the interesting complexities of doing this in 2 dimensions if the geometry is non-trivial. And he talks quite a lot about some quite interesting aspects of the dynamics and the regulation of this that, again, have not been followed up a lot. Unipolar regulation versus bipolar, this is still for the same gradient. How many different points you control. But really, I think what some of the most enjoyable kind of parts of the paper are his more general discussions about what he's really after. A feature of developmental processes, which is not often discussed, is the extent to which there are or will emerge general or universal principles which are applicable to development in the same way that there appears to be universal rules for genetics, or of more relevance for the transcription and translation of the genetic material at the molecular level. There's too often implicit embryological thinking that each step in development is a unique or special phenomenon with little general significance. I mean, I would say that in that side is where maybe he did have some of his biggest impact, because really, I think we do understand, and we do tend to be trying to look for general and universal principles in biology, and he was really one of the ones helping to push for that. So what was the most general part about his idea? I think it was really the fact that it's a 2-step process and that you can consider these 2 steps separately, specifying position informations versus interpreting position informations about values. It wasn't about it being necessarily morphogen gradients per se, or any particular molecular mechanism. It was about this separation into a 2-step process. This is why he then went to discuss the ideas of mixing flags with each other, which is a bit like mixing parts of different embryos with each other. So that you could have the French flag here and the stars and stripes. And if you put a bit of the stars and stripes organism into the field of the French flag, it would nevertheless know where it was, and it would grow, you know, a bit of the stars and stripes. This would be something like growing antennae or legs in the place of antennae or things like that in a real embryological experiment. Were there testable predictions, or was it just rather abstract? Well, in fact, again, in limb development, which was one area that he worked in fact, he did use it to make testable predictions, concrete predictions about if once the idea of a ZPA, a particular signaling region, was known, one could make predictions, purely sort of theoretical predictions, about what would happen to the arrangement of digits if you put the ZPA in different positions. And again, without going through the details, he used it quite genuinely as a testable prediction. And because one of the criticisms sometimes of position information is that it's almost not falsifiable, because this idea that it's almost a tautology. But in some of his own papers, he actually pointed out examples where predictions from this model didn't seem to turn out to be true. So it was falsifiable. So he was able to use it to make actual falsifiable predictions, and use it in a quite rigorous scientific way in that respect. So I think I want to go back now to just a bit to the more general thing about, that what his first paper could have done and was trying to do was a very old, ancient, classic distinction that is very important in science, and especially in biology. The difference between models and problems, it's a bit like the difference between the structure of something and the function of something, or the mechanism versus the behavior of something, or the genotype versus the phenotype, which could be the details of a gene circuit versus the pattern or morphology that it creates. This is a fundamental, distinction that we should not mix up. And yet, I feel that we did to a certain extent. So I've shown these examples here, two different models. These are the three parts that are in his very first paper from 1968. Two very different answers to one question. This is the question, and these are two different possible answers. I've now shown you more to show that there are other possible answers, like phase shift model is a different kind of model. It could also make a French flag. What about other kinds of problems? Well, a periodic pattern, which you call the stripy pattern problem, although it looks like a certain flag. This is a different kind of pattern, and so this is a different kind of problem. What are the different ways of making a periodic pattern? Well, I've already mentioned self-organizing reaction diffusion systems like a Turing model can make that. But it is also technically true. A morphogen gradient can make that type of problem, that kind of pattern. And therefore, probably almost any of these models could make it. The balancing model, I'm not sure if it would actually work. I haven't decided to try and work that one out. But again, in the answers or the models or the hypotheses, we've got two quite different categories. Ones that really are positional information, where each cell or each region could have different information about where it was, versus the more locally self-organized mechanisms that where cells don't necessarily know where they are, they're just responding to their neighbors in a very immediate way. So this is, what there really was, a distinction between problems, which was the French flag, and models, which were ways of solving it. So what actually happened? Why did we, what happened subsequently in the decades? Well, one thing that is undeniable is that in limb development, which was the field that he was working in, as well as others, evidence came, amazingly enough, in the same year that the French flag problem was published. Evidence came for explaining some aspects of limb development, the way that the digits are specifying. An explanation that is very much a positional information one, which is that if you take the limb bud of this chick wing, but much, much earlier than this photo when it's just a little bud, if you take the posterior region and graft it over onto the anterior region, you get this amazing mirror image duplication of the digits. You get 6 digits instead of 3, and they are organized in the opposite way. So it looks, to all intents and purposes, like a little French flag, where there was maybe some kind of gradient, and now there's 2 gradients in opposite directions. That was very exciting, and of course, exciting things, get a lot of attention, as they should. So a kind of an emphasis on gradient models, came up at that point in Lewis Wolpert's own work. But I actually was curious about this in a slightly kind of, over obsessive way. The more I was reading about it, is that if Wolpert himself never said French flag model, if he never said that phrase, how did it actually happen? When did it happen? And how did it happen? So I've been through all the papers. I did quite some analysis to look for, this is publications per year, and this is the number of publications that have the phrase French flag problem versus the ones that say French flag model. Now, his paper was in 1968, so that's the first little kind of blip there. Basically, for at least a decade or so, there were a number of papers referring to the French flag problem. They were citing his paper. Then, in fact, there's a period when it's not even much cited or discussed at all. And then around 2000 or so, it all kind of shoots up. But this time, everyone's saying French flag model. Well, not everyone, but a lot of people. But it is notable that there was one paper already back in 72, or was it 73? I can't remember. 73 that used this phrase. This is the first paper that actually says French flag model. The interesting, ironic thing is that the sentence in which this statement is embedded is saying exactly what I'm telling you. which is the idea of positional information and the French flag model, are really statements of what a pattern specifying mechanism has to achieve, not how it achieves it, not the actual mechanism, but what is the goal. It's the flag pattern, not the mechanism. But for some reason, which I have no idea why, this author put the word model, French flag model. Anyway, that was the first time that it was actually stated. But I mean, for another 2 decades, it was hardly used, that phrase. Whereas much later, you know, these are just examples of which there are hundreds. Like this, in the early and influential French flag model for the patterning of cells, a signal gradient across the fields is converted into gene expression domains by the concentration-specific response of target genes. I know that I'm over-laboring the point, but I hope that you see that this is not true. What was a gradient model was proposed to solve the French flag problem. But this is just stated over and over again, and I've probably stated it myself in my career. The models are therefore similar to the French flag model, in which the morphogen gradient defines gene expression boundaries via thresholds, etc., etc. So, you know, it's kind of interesting. I'm not criticizing anyone. I'm sure I've done the same thing, but, you know, something did get twisted if you compare to when you go back to his original papers. For a start, the French flag problem was not a gradient model. It was a problem, and it had multiple different solutions. But secondly, even, you know, as I've also tried to highlight, Lewis Wolpert was talking about all kinds of things, not just concentration gradients, but, you know, phase contrast gradients, all kinds of things. But we've kind of simplified it down to this caricature of what he said in his first papers. So in my work, just to go back to one slide about the work in our lab, I mean, we've done quite a bit of work trying to, in a sense, bring together the idea of non-positional information self-organized systems like Turing, but then being constrained by positional information, by localized expression of genes and signaling molecules to actually produce the results that is wanted. And so in this case, we actually believe that, for example, this is sort of the evolution of the model. The digit patterning is almost certainly an intrinsic mixture of a self-organized system versus with the necessary input for positional information as well, that the two things are necessarily linked. Doesn't mean that they're the same in any way. Doesn't mean they're versions of each other. They are two very different concepts that have to work together to get the right result. Both essential and both very different, but they're not exclusive. They have to work together. Let me see. I've got some slides I could go through relatively fast, and maybe people have questions on them about that. Jeremy Green and myself spent a little while thinking about the different ways that these two things, reaction diffusion or self-organization versus positional information, can work together. And essentially, you can think of at least 3 modes. We think of 3 modes. One is that reaction diffusion is upstream of positional information, meaning that you make a sort of periodic pattern, but then at a smaller scale, at a sub-level, this can act as positional information. Or the 2 things in parallel. So here we've got a gradient of some kind giving positional information, and here we've got a periodic system, and the cells sort of interpret both almost sort of at the same time to make their decisions. Or the last one, which is the one that I kind of was showing in the simulations, is where reaction diffusion system is actually downstream of positional information, where the positional information, rather than specifying cell fates, it specifies parameters in a reaction diffusion system so that you get different wavelengths at different places. So it's not that one end of the French flag gives a certain cell type and another end gives a different cell type. Both ends give both cell types, but it influences the patterning in this more interesting way. And maybe this explains some of the mutants that we see. But I just want to summarize by going back here. So I hope you have seen now that I believe that positional information, although Lewis Wolpert himself was very modest about what it's achieving, he was saying in some sense it's just reformulating things that are already known, but in a more clear framework. But it's not simply a tautology. It has been used to make testable and falsifiable predictions. It is a useful category, distinct category of mechanism, because it's distinct from self-organizing systems. And these are my final conclusions, take-home messages. It has simulated concrete testable hypotheses. I think it has a clear underlying principle. It's a separation of creating the information versus interpreting the information. That's really the key idea. Then the French flag is sadly simply not a model. It's not a mechanism. It's not a way. It's a question. It's a problem. And that's why I sort of almost feel sad about it as well, that flags, as I tried to emphasize earlier, is the almost perfect metaphor for something that's an abstract problem divorced from the solution, the mechanism. And yet we have completely associated French flag with an actual mechanism. In that respect, it's a great example of defining your question clearly before trying to solve it. And possibly because we sort of lost what I think what his paper could have been, something to help us keep them separate and therefore keep our minds more open to looking for different mechanisms like non-positional information mechanisms. This is the irony, that actual paper could have been the inspiration for looking for self-organized mechanisms. He even provided a self-organized mechanism in the paper. And yet we somehow slipped into a different way of looking at things. So I would like to thank my lab for all the wonderful amounts of work that I largely haven't talked about. And oh my goodness, I think I haven't updated this slide. Well, this is a little bit out of date, but yes, this is the new EMBL outstation, the new EMBL unit in Barcelona, part of EMBL. We have 5 group leaders at the moment, and we're actually at the moment just going through another recruitment round. So thank you very much for your attention.
Great, thanks a lot, James. That's fantastic. Food for long thought thereafter. It's actually interesting, I think, that Lewis himself didn't help this, because in his latter years, he kept on talking about the French flag model himself and many things. And I think he propagated it rather a lot, and including in some interviews which are available online that are very nice, but where he talks about his own life and a number of episodes. And he talks about the French flag model there, not problem, which is very interesting. Made many years ago, towards the beginning of your upwards curve, I think is when they were recorded. So I think he didn't.
I have to admit, so I've looked into that as well. There are, I think, 3 sort of, or 4 like reviews / interview written published ones in which the phrase exists and he's listed as either the interviewee or author, co-author. Mm-hmm. And that did happen, but actually it was like more than 30 years was the first one. Yeah. After. So for 30 years, and Lewis has published many papers in those 30 years. For 30 years, he did not write French flag model, even though that phrase had started becoming common in the literature. But then at the very end, he did indeed slip. It's true.
Any other quick? Chris Barnes has a question.
Hi, James. Thanks for the talk. It was really interesting. I just wanted to ask about, you say that the positional information model and the self-organization model are 2 separate things. But is that because of the way we view the system, or because ultimately, the gradient in positional information could be could arise through self-organization as well. So I'm just interested why you think they're two fundamentally different mechanisms.
Yeah. So, I mean, that's quite there are many things that can be said about that. I'll start by putting Lewis Wolpert's own words, okay? Since I am, I've been accused of being anti-Wolpertian, and I want to show that I'm actually, I think he was a genius. This is what he wrote about this balancing model. He said, this type of model may be contrasted with the previous one. Not only can a cell never compute where it is in the system, but the system has no gradient. So that's at least one factual, practical difference. I mean, the idea of positional information is that you have a monotonic distribution of something, of a variable. And as I said, it doesn't have to be a concentration variable. It doesn't have to do with signaling intensity per se. It can be to do with the phase of an oscillation or the frequency of an oscillation or anything. It can be any variable, a scalar variable, distributed monotonically across space. That is at the heart of positional information. And this other type here, there is no gradient of anything, of concentration, of phases, of oscillations, of ages. I mean, another one that's actually, that I forgot to mention, not to overload things, but you can have positional information based on a gradient of age, for example. Like the classic progress zone model from Lewis Wolpert and Julian Lewis from 1975, I think, is also positional information, not based on gradients of concentrations or oscillations or anything, but gradients of sort of the time that a cell has been out of influence from a signal. So, I mean, this is a sort of technical difference. The self-organizing models do not have a gradient of some variable across space, which is instructing the cells what to do. It doesn't mean that that's about the concept of what the self-organizing models are. What Jeremy Green and I were trying to illustrate in these slides here is that, of course, you can have a system that does both. So you can have a system here where the cells have access to a periodic information over space and also a monotonic distribution over space, and they can use both of those to make a bunch of decisions. That doesn't mean that this is somehow the same as this. They are very different. And there are a bunch of other differences as well. I mean, in the Turing, kind of pure Turing type of idea, what those are capable of doing is genuine symmetry breaking. So in a system where there is no apparent symmetry apart from noise, they can genuinely break symmetry and form something organized, whereas positional information systems, by definition, cannot do that. I mean, in that respect, this isn't surprising. I mean, positional information systems are an abstract theoretical concept, and it's defined in a certain way, and the way it's defined is such that they cannot break symmetry by definition. I mean, we don't even have to look at biology for that, because again, that's the thing. These are sort of abstract concepts. What we actually find in real messy biology is a whole other thing, but the concepts are completely distinct.
Okay, I take your point from the cell point of view, yeah, the positional point of view, yeah, knowing where the cell understands its position, but I guess ultimately they follow the same mathematical equations. That's kind of my point. Not
really. I mean, so the self-organization, yeah, I mean, there's a whole bunch of, in a way, self-organization systems are more, in a way, mathematical. Let's say there are mathematical proofs about what makes a system act as a Turing system or not act as a Turing system, and you can define it quite mathematically, and we've done some of that work as well. In some sense, a position information system is almost too simple to even be mathematical. I mean, it's just a statement of saying that if something is different in different places, then the cells can respond differently in different places. I mean, that's why it's been criticized as a tautology. If you say it the wrong way, it sounds meaningless. Like X is different across space, and the cells can sense X, so then every cell can be different. I mean, if you state it that simplistically, then it almost seems a meaningless concept, and that has been that criticism has been leveled at it. But here I'm more, highlighting that to point out that it's almost not a mathematical thing. It's just a sort of very simple, common sense thing in a way.
Okay, thanks.
Roberto has a question.
Very nice talk, James. I mean, I agree with the comment that Claudio made that I think probably Lewis was in part responsible for this confusion that you described. But I remember talking with him at relatively recently. He was very against the gradient as a mechanism because he thought it was very messy, very unreliable. But not for the reason that you described. I mean, it was more, the problem was in the mechanism, how a gradient can generate a reliable pattern. But I agree with Claudio that he probably was a bit responsible in this, at least not clarifying this confusion as you have done it. I have don't you think that, related to the question that Chris raised, considering biology and as you said, that how messy the biological systems are, do you think it's more likely that you could, the cell respond not with one mechanism, but with a variety of different mechanisms? And these are maybe just two examples of the many mechanisms that the cells can use to generate a pattern.
Absolutely. So this is in a sense what I'm trying to highlight here, if I can bring it up again. Okay, so the point here is that to make this pattern, you could do any of these mechanisms, and maybe you can do all of them at the same time. To make this pattern, there's many different mechanisms that could make this pattern. And why would biology, there's no laws of biology that would say, well, we're only going to use one mechanism. I think it's true to say, and I didn't emphasize this much because it was sort of not the main point, but I think it's true that certain mechanisms look more logical, let's say, or more well suited for making certain kinds of patterns. So, the periodic pattern is not very well made exclusively by a morphogen gradient, from a theoretical point of view, but it is possible. And so, I couldn't agree more with what you say. Real biology, may be well represented in this diagram, which is to say, if you want to make, head here and your tail here, and there's different ways that you can do it, maybe you combine a bunch of them simultaneously to do it. Absolutely. So this is not in any way against that. Going back a little bit also to your first question and the one here, as Claudio mentioned at the beginning, I kind of grew up as a computer geek and rather theoretically minded, and my bias is ideas and the concepts, and the ideas and concepts, are not the messy biology, which is their weakness, but they also not the messy biology, which is their advantage, which is to say, the ideas are defined by us. And so it's particularly disappointing if we mix up our own ideas. Mixing up the biology is fine because the biology is a total mess. But at least when we talk about our concepts, we should understand which concept means what. Now, how the concepts map into the real biology, probably it's a mixture of lots and lots of things. And, again, in terms of having multiple mechanisms in one system, this is exactly what Jeremy Green and I were trying to talk about here, having multiple mechanisms in one system simultaneously and talking about the different ways it works, but still acknowledging that the ideas are different ideas, but they can work together. They can be either add different features, or they could even work together to just back each other up, which are also two differences. Here, the periodic system is responsible for the periodicity of the digits, and the gradient is responsible for the fact that the digits have different identities. So the gradient is responsible for a different thing that the periodic mechanism is. They're working together, but they're responsible for different things simultaneously. But you could have the opposite, which is that indeed a positional information system and a self-organizing system work together simply as a backup to achieve the same thing in a different context. For example, boundary sharpening. Boundary sharpening is a very self-organized type of thing typically, because you essentially have communication between the fuzziness of the two boundaries, and they reinforce each other, and you have positive feedback, and it ends up making a sort of a bistable switch kind of thing. So there are many ways that these different principles can work together, but it doesn't mean that they're the same thing, theoretically.
Yeah, I completely agree. Can I ask you just a short question? I mean, out of curiosity, because you have been studying, or you have looked at the citations of Lewis Wolpert paper. So what happened in 2000 that there was this peak of citations?
Yes, indeed. I should mention more about that. I mean, this was really just when a whole bunch of experimental evidence of systems, especially in Drosophila, started being just sort of, stated correctly as that, bicoid, things like, bicoid as a real gradient and controlling, the gap genes and controlling the payroll genes and all of that, and then in a few other model systems as well. It's basically when really the experimental evidence for a bunch of model systems started to become, really, really incontrovertible, and that they do work as concentration gradients. So it's another point that I should have really mentioned. It sounds like I'm almost maybe arguing that concentration gradients are not so important. I mean, they do seem to have been found a lot, and I think they are extremely important. And that is a lot of the reason why we have focused on those as mechanisms. It's just a shame to me that we attached French flag onto that mechanism, because French flag is an equally useful phrase for, as Lewis Wolpert himself showed, for a non-gradient mechanism. This mechanism. This is not a gradient mechanism, but it solves the French flag. So all those bicoid gradient mechanisms in Drosophila and all the other ones in all the other systems, whatever, retinoic acid or sonic hedgehog or whatever, they just shouldn't be called French flag models, in my humble opinion.
So can I go back to a question from Roberto's previous question and your answer to it, and just say something that you may or may not disagree with this, but when you look at the idea that multiple mechanisms can cooperate to generate an outcome like a pattern, the fire alarm's going off in the building, so I know we're all kind of sorry about the background noise, but we may all explode in a minute. Anyway, so two things. First of all, do you agree that in part the view that you have one dominant mechanism for everything, or even in some cases one gene for a function, is partly the fault of genetics, in a sense? Is that people using genetics to decide whether something is important or not on the basis of whether it's necessary and sufficient, perhaps, or at least necessary for a process to take place. And if it's not necessary, it cannot be indispensable, therefore this is not the mechanism. And that's been a dominant way of testing things. But at the same time, genetics is also very powerful in terms of providing ways that you can design experiments that are almost like a computer program, in that you can do beautifully designed, especially somatic cell genetics experiments or combinations, where you can look at interactions between things that uncover rules. So there's a big contradiction there. Now, Lewis himself said to me many, many times that he didn't believe in redundancy. And the reason why he didn't believe in redundancy, almost verbatim, this is what he said, is because evolution would never allow that to be perpetuated. Because he thought that if there are overlapping mechanisms that achieve the same end, then one of them will drift out through evolution. There is no double assurance. And that's the way he kept on repeating that. And on the other hand, you're saying something different, right? Because you're saying that there are different aspects of the pattern that get conserved. And Roberto, we had this discussion yesterday also in another seminar. Roberto's work himself has shown that there are multiple mechanisms that all seem to give you the same outcome when you perturb them, but they all affect collective cell migration of the neural crest.
Yeah. Well, you know, it's funny that you point out what you say Lewis Wolpert mentioned many times, because I also remember him saying something a classic Lewis Wolpert quote was, for all of these knockouts that didn't seem to cause a phenotype, he said, and I'm sure you'll remember this quote as well, but have you taken the mouse to the opera? Because I mean, the point was, and it's it's the opposite point of the one he made to you as well, that we are testing these systems under such abnormal laboratory conditions and with such blunt instruments for actually telling whether something's normal or not. I mean, my instinct would be that it's actually more about the fact that we see so many self-correcting behaviors in systems. Like if something's 5% too big, it can be adjusted. If a gene expression pattern is a little bit too high, it can go down. I mean, self-adjustment, homeostasis, or appears to be, I think, I don't think there's any question that it's there in so many developmental processes. Now, that fact alone, I would say, shows that evolution is being selected to be able to correct problems from noise, problems from thermal fluctuations, from whatever. I mean, an embryo is never in a perfect hermetically sealed box from the outside world. It has to cope with some variation, temperature, nutrients, whatever. So that slightly argues against, I think, the thing that he was mentioning to you, because a backup mechanism may look redundant in laboratory conditions, but under 1 million years of evolution of coping with every kind of fluctuation you can think of, the backup mechanism will have enough selective advantage to be kept in place. How many embryos, what percentage of actual embryos of a given species would it need to save to have an evolutionary selective advantage? Quite a small proportion. So I don't know, but I also, I mean, my suspicion is that in a sense, that there can be backups, but I actually believe it's probably a bit different than that. And this is ultimately what I'm more interested in, is that I think it's very important. So what I've been saying today, I think is important, which is to understand the concepts that we discuss independently of how they apply to biology, and then apply them to biology. We've got to be clear on a given model, what it means, what it says, what it doesn't say. But having said that, I do suspect that real mechanisms in real organisms and embryos are much more or much less separable into neat things like, along this axis you do this, and along that axis you do that. Biology just has to make things work, and I think that rather than redundancy, I think you'd have there exists what I would call distributed control. So many different things there, and the control of any given feature that we choose to measure, and it's our choice. It's like the observer subjective issue there. What thing of an embryo do we choose as an important thing? So we choose something, we measure that thing, and then we see which things interact with it, which things affect it. But I think that actually the control of any given observable will be very much distributed across different things that we call different mechanisms. So you could say that there's different mechanisms that are backing each other up, but maybe it's going to turn out to be more true to say we have this kind of big mess of mechanisms that just somehow all work together, and they all manage to get all these different things working, like the length right, the width right, the proportions right, the size, proportionality right, and it's a bit more of a kind of an emergent thing. So despite everything I've just been saying about this mechanism, this mechanism, I do think it's important from an intellectual point of view to understand what we mean by a certain thing and to keep them separate. But I do also think that the real biology is not likely to have a pure Wolpertian positional information system in any given example. It's going to be actually a sort of slightly maybe difficult to understand mixture of dynamical processes going on at the same time. And through a certain prism, it can look like that. But through a different prism, the same thing might look like a different kind of system. And it may not even be because the different things are overlaid on top of each other, but rather that it's actually a more complex thing, and it just looks like that depending on the way that we look at it. That's a bit fuzzy, I'm afraid. Oh, you're muted still, Claudio.
Sorry, I think we've gone a bit over time, and some people are leaving partly because of the fire alarm, and partly which is now stopped, and partly because of other things. So maybe we should end the